Organoids, or the organoids that we at Hub Organoids use, are adult stem cell derived cultures. That means that we can take a biopsy or a sample from a patient and take that to the lab, and we grow it out into organoid cultures, which we can then put in the freezer and we can use as a platform for research and drug development. Organoids, of course, as a new technology, are in itself quite expensive. On the other side, the drug cost, there we have to see how it works. I think it is an expansion of using organoids. We want to bridge to the clinic, stratify patients easier so the clinical trial becomes cheaper. And so all these steps, we're really early on. We really think that will have an impact in the coming years.
A lot of the challenges we're seeing in precision medicine is reproducibility and scale. So how do we make a good model for a patient and then benchmark it and then translate that to other patients? So what are the readouts that's actually predictive of that patient's response? How do we look at a patient's organoid or a model organ in the dish and say it's responding like the patient would? And then if we do get that, how do we do that for every single patient or bulks of populations at scale?
For our center in particular, we're limited in terms of the availability of tissue. So for small cell lung cancer, many of those patients do not receive a surgical resection when they present with disease. They go straight into standard of care chemotherapy. So we're generating these tumors kind of from scratch by taking established cell lines in the SCLC community, putting them into our organoid models, and then creating these miniature tumors from there. So that's a big challenge.
In the next 10 years, we're starting to develop these umbrella trial protocols and the ability to specifically target individual tumors. But the real need is to show that our modeling approaches are really able to say, this drug is the right drug for this patient, and thereby build out our ability to take the data that we're able to generate now at high throughput for individual models and apply it to individual patients. And so that's really the key barrier that I think correlative studies and the ability to do co-clinical trials will expand and allow us to sort of step into in the next 10 years.